Hi friends, today we are going to see the past year NEET questions for the chapter Cell Cycle and Cell Divisions. In this video, we will cover part 3 today. So earlier we have completed 2 parts, so kindly have a go through. Okay, now let's start with the first question here today. During cell division in apical meristem, the nuclear membrane appears in dash. This question was asked in the year 1997. So before we go directly into the answer, you should know what is meristem. Right? Meristem are the regions in plants in which mitosis will take place. Now when I say apical meristem, it denotes to the tips. Either it can be the root tip or the shoot tip. Either of this. Okay, meristem are the region in plants in which mitosis take place, and apical meristems denote to the tip of the shoot or the root. And what is the function? If you see, they contribute to the length of the root or the shoot. Okay. Apart from that, what you have to know here is apical meristem, they can denote either to a root or a shoot, as I already told. See, length increase can either be towards a root hair, it can go or penetrate into the soil. So this shoot system will talk about the height of the plant. Now coming back to the question here, during cell division, the apical meristem, the nuclear membrane appears in dash. So you have 4 different phases given here. What happens in each phase we will discuss now. Metaphase. When we talk about metaphase, we talk about the appearance of the spindle fibers. Right? Appearance of spindle fibers. So this cannot be the answer. What happens in anaphase? Anaphase involves the movement of chromatids towards the poles. So it is not the right answer. Cytokinesis marks the division of the cytoplasm of the parent cell into the daughter cell. So this is also not the answer. The correct answer is telophase. Option C is the right answer. Now to your right side, you can see a clear image of the mitosis and the cell division which happens in a normal eukaryotic cell. It starts initially with interphase and slowly it ends with cytokinesis. So during cell division in apical meristem, the nuclear membrane appears in telophase. Option C is the answer. Now let's move to the next question here. This question was asked in the year 1996. Colchicine is employed to dipolarize a haploid cell as it dash. So what does colchicine do? We have already spoke about colchicine in the earlier parts. In part 1 and part 2 we had discussed about it. See, the natural work or the natural function of colchicine is to inhibit. Okay, it restricts. It is also called as mitotic poison. Okay, now colchicine is also used as medication to inhibit the or to stop the cell cycle, mitotic cell cycle. Now coming back to the options given here. Look at the first option, it inhibits mitosis. Option B inhibits the formation of mitotic spindle. The correct answer goes to option B because colchicine inhibits the formation of spindle fibers. Therefore, the correct answer is option B. See, in the solution part they have clearly told you colchicine is a mitotic poison which inhibits the formation of spindle fibers. Colchicine is also used for a particular disease called Gout's disease. Okay, Gout's disease. Now this disease, if you see, it is actually in resemblance with, it is also called as Boucher's disease, and this disease they use this particular colchicine. It is also used as a steroid actually. So these are just extra information that you need to know. The main function of colchicine is it works as an inhibitor. Therefore, the correct answer to this question is option B. Now let's move to the next question here. This was asked in the year 1994. Best stage to observe shape, size, number and chromosomes is. So once again, this is a very easy question. Now, when we need to observe the number of chromosomes. Now what is chromosome? How can you define chromosomes? Chromosomes are the ones which carry the genetic material. We all agree with that, right? Apart from that, you can also describe chromosome is DNA. Yes, it carries genetic material. Therefore, it is the DNA molecule with part, or either it can contain a part of it or it will contain the whole genetic material of the organism. Most eukaryotic chromosomes include packaging of proteins which are aided by the chaperone proteins bind to it and condense the DNA molecule to prevent it from becoming an unmanageable tangle. Therefore, this is all about chromosomes. Now, when we have to observe, okay, you need the distinct or you can call it as prominent. Okay, prominent. Only when the chromosome is distinct or prominently present, only then you can count it. During metaphase, the centromeres of the chromosome lies on the equatorial plate according to their size and the spatial arrangement. Therefore, the best time to, or the best stage to observe size, shape and number of chromosome is during the metaphase. Okay, while rest of the phase, if you see interphase, prophase or telophase, you will have the chromosome, but they might be in a condensed form. Okay, they might be in the condensed, or you can say it is in the coiled form. Therefore, the correct answer to this question is option B, metaphase. So here you can see the diagram to your right side, which clearly shows you chromosomes line up along the metaphase plate. So what happens in the metaphase, they have shown you in this diagram. So let's move to the next question. This question was asked in the year 1992. Number of chromatids in metaphase or at metaphase is dash. Easy question again. All you need is a little logic, right? Fine. In both mitotic and meiotic metaphase, the number of chromatids per chromosome will be 2. So I've given you the clue, it is 2 now. So what will be the answer now? Now first tell me, what is chromatid? What is the difference between chromosome and chromatid? Chromosome we already saw in the earlier slide. Now chromatid is also a chromosome that has been newly copied. So you can just remember one part here, which is newly copied. We already saw in the earlier parts that in the end or in the last part of the life cycle, 2 daughter chromatids will be formed. Right, 2 daughter chromatids will be formed. Now what is it actually? They are the copies of the earlier chromosome. They are copy of the initial chromosome. Chromatid is a chromosome that has been newly copied, or the copy of such a chromosome of 2 of them still joined to a original chromosome by a single centromere. Before replication, one chromosome is composed of one DNA molecule. I'm talking about the before replication. So here therefore the correct answer is, already I've given you a clue. So what will be there? Option A is the right answer. 2 each in mitosis and meiosis. See, in mitosis and meiosis it will be same. There will not be any differences. If there's differences, then the cycle has said to be, it has malfunction. Therefore the correct answer to this question is option A. Okay, now let's move to the next question here. This question was asked in the year 1991. Mitotic anaphase differs from metaphase in possessing dash. Again, this is an easy question. You only need a little logic. Okay, now mitotic anaphase is where the sister chromatids will separate from each other, right? And they begin to move towards the opposite of their poles. So what happens here? I've clearly told you the sister chromatids will separate. They will get separated, and that is, they get separated from each other and they will move towards the poles. Okay, they will move towards the poles. Now when they get separated, it is not going to have any change in the chromosome number. Okay, therefore it is the same number of chromosomes. But how about the chromatids now? What will happen to the number of chromatids? Will it remain the same? No, it will be reduced, or it will get reduced to half the number of chromatids. Why it should reduce? Imagine the number of chromosomes is going to double every time, every replication, or during every life cycle or every cell cycle. Then you will call that as a malfunction, because the concept is very simple, where the n will get converted into 2n and 2n will again convert it into n. So diploid getting into haploid, haploid getting into diploid is a very common process. So the number of chromosomes will not change, but the number of chromatids, where we have the last part where 2 daughter chromatids will be formed, okay, that will be reduced to half. So mitotic anaphase differs from metaphase is that the number of chromosomes will be same, but the chromatid number will reduce to half. Therefore the correct answer to this question is option D. Okay, now let's move to the next question here. Fine, select the correct option. Okay, this is once again easy. You just need to recall what I spoke or what I taught in the earlier parts. So when you get these kind of questions, always remember to have or search for keywords so that you can easily answer. Now look at your A and the 1 and the 2. First one, synapses aligns homologous chromosomes. So here the keyword is it is homologous chromosomes. So whenever we talk about homologous chromosomes, we know either it should be bivalent or synapses. So we have got a clue here, and this takes place in the zygotine. Therefore the right answer for this is zygotine. So this is a keyword that you need to identify here. Now next one, synthesis of RNA and protein. Synthesis of RNA and protein takes place in which phase? G2 phase. Therefore this is for B. Action of the enzyme recombinase. In which one will it take? See, action of enzymes with the recombinase with the help of the recombinase enzyme is seen in pancreatine. Okay, pancreatine is where the crossing over is mediated. You need crossing over, right? So this crossing over will be mediated by the pancreatine, that is action of the enzyme recombinase. Next one, if you see, centromeres do not separate but chromatids move towards opposite poles. So that will happen during anaphase 1 or 2. It will happen during anaphase 1. Therefore D comes here. Therefore the correct answer to this question is option A. See, whatever I explained to you is what is given to your right side, so you don't have to get confused. Okay, option A is the right answer for this. Now look at the next question here. In meiosis, crossing over is initiated at dash. I think this is an easy question for you because we just now discussed about it, right? Now what do you mean by crossing over? Very important that you need to know that. Meiosis, we all know it is a process very important in cell cycle, and whenever we talk about this particular sequence, we always have to remember that when a single cell dies, the cell cycle will go into mitosis. Now when we talk about a complex process, or when we talk about a reproduction process, it is very well understood it's a tedious and it's a long process, it's a complex process. That will be taken care by meiosis. Meiosis is a process which was discovered by Oscar, where a single cell divides twice, okay, to produce 4 cells. A single cell will divide twice to produce 4 cells. We all know that. And that will contain half the original amount of the genetic information. These cells are our sex cells, where it is the sperms in males and eggs in female. Crossing over, now coming on to the concept, what do you mean by crossing over? Crossing over is a process by which 2 chromosomes exchange some distal part of the DNA. This process, if you see, will occur in the pancreatine. Okay, it occurs in the pancreatine stage. And which phase do you think it will occur? I've told you the answer. Option A is the answer. I've told it's pancreatine. This will occur in the prophase 1. Okay, prophase 1. Therefore the correct answer to this question is option A, pancreatine. Okay, now coming on to the next question. Choose the correct option for the following events of meiosis in correct sequence. Okay, you just have to find the order. So I will tell you the order now. First we start with synapsis. Okay, then we move into crossing over. Synapsis is what? The pairing of the homologous chromosomes. Crossing over is what? Just now we saw, right? Crossing over is a process by which 2 chromosomes will exchange some distal part of their DNA with one another. So once homologous pairing is done, then moves to crossing over. Then comes the terminalization of chymasta. So here if you have terminalization of chiasma. Okay, so this is 3. Next you have the disappearance of nucleolus. So what is the process now? It starts with synapsis, then it moves to crossing over, then it moves to terminalization of chiasmata, and then it ends with disappearance of nucleolus. Okay, so this is the order. Already we discussed what is synapses, what is crossing over. And chiasmata also we have discussed in the earlier parts. So this is the order that we follow. Therefore, the correct answer to this question is option A, B, A, C, T. Okay. Now next question here. This question was asked in the year 2015. The chromosome in which centromere is situated close to one end are. Okay. You're given 4 options here. Right? What do you mean by this centrics that are given here? Metacentric, submetacentric, telocentric, acronocentric. We'll discuss one by one in detail. Now when we see acronocentric, where one arm will be short. Okay, acronocentric is a acronocentric chromosome is where a chromosome in which the centromere is located quite near to one end of the chromosome. Usually humans will have 5 pairs of acronocentric chromosomes. Okay, but the people with Down syndrome will happen to have more than 5. That is why they have the Down syndrome. Now what do you mean by telocentric? I told you what is acronocentric. Okay, telocentric can be described as the centromere it is where the centromere is located at the terminal end. Okay, so this is one end and this is terminal end. Okay, so what do you mean by metacentric now? I've told you, I'm giving you a clue. I've told the answer in fact. Okay, now metacentric chromosome is where the chromosome will be in the middle. Okay, submetacentric, if you see, they will be somewhat in the middle but not exactly middle. So when you talk about one end, the correct answer goes to option A, acronocentric. So to your right side you can see a picture which tells you what is this. So here if you see metacentric, it is exactly in the middle. Submetacentric, it is not in the middle as well as not in the exact top, somewhere close to it. Acronocentric, it is to one end. Telocentric is towards the terminal. Therefore, the correct answer to this question is option A, acrocentric. Okay, next question here. This question was asked in the year 2014. Okay, let's read it here. The enzyme recombinase is required at which stage of meiosis? Easy question. We've already discussed an enzyme recombinase is required for which process? Any idea? Do you remember where did we see that? We saw in the match the following. If you correctly remember, we saw it in the match the following. Recombinase enzymes is required for crossing over. Always remember it, please don't forget it. Okay, now coming to the solution. See, the enzyme recombinase is required at the panctine stage of the meiosis. It catalyzes the exchange of short pieces of DNA between the 2 long DNA strands, particularly the exchange of the homologous region between the paired maternal and the paternal chromosomes. Therefore, the enzyme recombinase, as I already said, it is required for the crossing over. Whenever we talk about the maternal and the paternal, you always have to remember this stage will occur in the panctine. Okay, the correct answer to this question is option A, panctine. Okay, now let's move to the next question here. This question was asked in the year 2013. During meiosis I, we all know meiosis will happen as 2 parts, meiosis I and meiosis II, right? Fine. During meiosis I, the chromosome starts pairing at dash. Whenever we talk about pairing, you have to remember one thing. The pairing that they talk is in resemblance to the homologous chromosomes. And when we talk about homologous chromosomes, we all know we are talking about zygotene. Okay. During zygotene, the substage of the prophase 1, that is, we are talking about the meiosis I now. Okay. Chromosomes start pairing together and they call that process as synapsis. We already saw that. Such paired chromosomes are called as homologous chromosomes. A complex structure, that is synaptonemal complex, you call that as synaptonemal complex, is formed by a pair of synapsed homologous chromosome, which you can either call it as the name bivalent or you can call it as tetrad. What's the spelling of tetrad? I'm sorry, tetrad. Okay, therefore the correct answer to this question is option B, zygotine. Whenever they talk about meiosis and whenever they talk about pairing of chromosomes, you have to remember that they are talking about the chromosome pairing between homologous pairs. Therefore the correct answer to this question is option B. Next question. Given below is a representation of certain event at a particular stage of a type of cell division, and which is this stage? So you're given 4 options here. Okay, prophase 1 during meiosis, prophase 2 during meiosis, or it's prophase of meiosis, both prophase and metaphase. The correct answer is prophase 1 during meiosis. Why? The given diagram represents the crossing over. If you can clearly see, it talks about the crossing over that takes place in the panctine stage of the prophase 1 during meiosis 1. So you can clearly see here it is the crossing over. So you can see here this part, they are talking about the crossing over. So that's why this stage belongs to prophase 1 during meiosis. Okay, option A is the answer. Next question. During gamete formation, the enzyme recombinase will participate during, I think this is once again an easy question for you to identify. Okay, so any clue what is the answer? During gamete formation, the enzyme recombinase, we already saw. Recombinase enzyme is required for crossing over. Recombinase enzyme catalyzes the exchange of short pieces of DNA between 2 long DNA strands, particularly the exchange of homologous regions. Therefore, the correct answer is prophase 1. Okay, see, we saw this answer in detail in the match the following. Okay, recombinase enzymes is required for crossing over, and this stage is prominent in prophase 1. Therefore, the correct answer to this question is option C, prophase 1. Now, let's move to the next question here. Identify the meiotic. Okay, identify the meiotic stage in which the homologous chromosomes separate while the sister chromatids remain associated at their centromeres. Okay, you're given 4 phases now here. Metaphase 2, anaphase 2. So it is somewhere in the metaphase or the anaphase. That is why they have given you. But you have to clearly know whether it is 1 or 2. Okay, metaphase, you know, it is the alignment of the spindle fibers. So it will not come under metaphase now. Now you just have to identify whether it is anaphase 1 or 2. The correct answer is anaphase 1. Anaphase 1 begins with the 2 chromosomes of each bivalent. That is, we are talking about tetrad. Right, we are talking about tetrad here. So what happens here? They separate and they start moving towards the opposite poles of the cell as a result of the action of the spindle fiber. In anaphase 1, the sister chromatids remain associated at their centromeres and they move towards the, that is, though they remain associated with the sister chromate, they move together towards the poles. Okay, they don't get separated and they move. All together they will move towards the poles. Therefore, the correct answer to this question is option C, anaphase 1. Okay, now let's move to the next question here. So this is the last question for this part 3. Genetic map is one that dash. Asked in the year 2003. So what is a genetic map actually? Okay, what is a common work or the function of a map? What does a map do? It helps you find something, right? Likewise, what does a genetic map do? Genetic mapping or genetic map describes the methods that is used to identify the locus of the gene. Very, very important. It is used to find the locus of the gene. See, the normal map that we use to find places, or it might be any destinations, we use a map, right? When we are traveling, we don't know the direction, then we use a map. So likewise, genetic mapping is used to find the locus of a gene, where the gene is exactly located, the destination, and the distances between the genes. The essence of all genome mapping is to place a collection of molecular markers onto their respective positions of the genome. So molecular markers, can you if you see, they come in different forms. Okay, now here the question is that what does a genetic map do? Is one in that So first option, it shows the distribution of various species in a region. Wrong. They are talking about the general function of a map. Second one, establish the sites of genes on the chromosome. Third one, establishes the various stages in a gene evolution. Wrong. We don't talk about evolution here. We only talk about the current status. Therefore, evolution doesn't come here. Option A is also wrong. Shows the stages during cell division. No. The correct answer is option B. As I already said, a genetic mapping or map is used to find the locus of a particular gene, and you can also find the distance between the genes so that you can easily locate and target to do any action. Okay, either you target to destroy it or you target to rectify it. It can be anything. Therefore, the correct answer to this question is option B. Okay, so with this we complete part 3, cell cycle and cell division. So let's meet in the next part. Thank you for watching my video.